All right, in this last video for this module, we will talk about high-throughput sequencing as a type of molecular marker. And in my opinion, we've saved the best for last, not to be biased, but high-throughput sequencing I think is super cool and it's probably my favorite marker. And one of the reasons why I really love high-throughput sequencing and especially why it's so beneficial for my personal research is that you can do high throughput sequencing on non-model organisms. So that a non-model organism is something like what's shown in this image on the right. So an organism like a sperm whale or a kangaroo rat or a red-eared slider, that turtle. In contrast, a model organism is something like a fruit fly or a nematode, or a mouse. Model organisms are something that people have studied for a very long time, but mostly just as a way to study like human associated questions, for instance. So Drosophila and mice are often used to do molecular research type questions that are focused on coming up with some kind of human-based application. Whereas non-model organisms, like the ones shown on the right, they're considered non-model because we don't have as much genetic information about them. And so in the past, having limited genetic information could limit the types of studies you could do, especially in a molecular ecology sense. But because of the way that high-throughput sequencing works, you can actually do these types of studies on any organism, including these non-model ones, and so it's really ideal for these molecular ecology questions where often we're just interested in what this particular organism is doing, not for any human-based purpose. So before we get into it, I want you to reflect and think about what are the different types of high-throughput sequencing. So we've mentioned one in a previous video, and maybe from your reading you remember a few. So remember, the more you practice recalling things and pulling them out of your brain, the better you'll remember them. So first, think about what different types of high throughput sequencing are there. So in this video, we'll talk about 3 different types. The first being what we call reduced representation. The second being targeted. And the third, whole genome sequencing, which is often abbreviated WGS. And so just as a quick overview of how these types of sequencing are different, say we have this hypothetical stretch of genome here. So that's our entire genome. Reduced representation, what we're doing is sequencing little parts of that genome, but spread throughout the entire genome. Targeted, what we're doing is sequencing one particular region only out of the entire genome, but we're sequencing that one region a lot. And then whole genome sequencing, we just sequence the whole thing. So that's a general overview of how these different types of sequencing are different, and we'll go into the details in the rest of the video. So we'll start with the first one, reduced representation sequencing. And what this does is it generates large amounts of sequence data. So again, any high throughput sequencing technique is going to give you lots of data. But you're getting this data from a subset of the total genome. So that's where this reduced representation name comes from, is you're looking at a subset of the genome. Oftentimes what that means is that you're breaking up that genome into smaller fragments and then only sequencing some portion of those fragments. One of the ways that you can break up that genome is using restriction enzymes, which you may have used in some of your lab classes in the past. So what you can do is break up the whole genome using these restriction enzymes and then only sequence some of that with the high throughput sequencing. And that's actually what's used in the main example for reduced representation sequencing that we'll talk about, and that makes sense from the name because they use restriction enzymes. Restriction site associated DNA sequencing, or RAD-seq for short. And we've actually talked about this type of sequencing a little bit before. This is what I've used in that study with the snails that I've been referencing throughout this module. So RAD-seq is one example of reduced representation sequencing. And I'm going to walk us through some of the details for how this sequencing works. So bear with me here, I'll walk us through this. So RAD-seq again is that type of reduced representation sequencing. And when you do a RAD-seq study, because of the way the technology works, what you end up with are many sequence reads, so they're called reads, and they're really short. And so because they're short, that means they don't have the full sequence of the gene. And so what we need to do is use these bioinformatics data programs to then assemble those small reads into contigs, which are these longer. So reads are really short, and contigs are longer, ideally representing one gene. And so if we walk through this cartoon image here, so we start with our raw reads, which are really short. So everything above this line here, those are our short raw reads. We then use bioinformatics data programs to figure out where some of these reads overlap. So that's what's shown here. You're almost like Lego blocks where you're stacking them together. So you're figuring out where these short reads overlap and then collapsing that into these single sequences representative of these different regions of the genome. So these contigs are, again, those longer sequences that you're reconstructing from those shorter reads. In this hypothetical example, the data that we obtained is from 3 different regions of the genome, a purple, green, and red region. So when you're working with RAD-seq, your goal is to assemble reads that are short into these longer contigs. And then what you can do, as we've talked about before, is identify in each of those contigs, or sometimes they're called loci or a locus. Within each of those assembled contigs, you can then identify SNPs. So in RAD-seq, really that's one of your ultimate goals, is to identify many SNPs that are spread throughout the genome of the organism. And as we talked about before, if you're interested in knowing how individuals or what SNPs help an organism maintain its fitness in different environments, you can identify specifically adaptive SNPs. So this is a table from one of my studies. Don't worry about these columns, but on the left we have these different loci listed. So this is essentially different contigs that we've then identified SNPs in. And by comparing our sequences to known databases, we can figure out what particular regions of the genome these different SNPs are in. So that's this abbreviated description column here on the right. And what I have highlighted in yellow, all of those particular genes are involved in something called stress granules, which we'll talk more about later in this semester, but they help organisms survive stress. So by doing this RAD-seq study, what I've been able to do is identify these adaptive SNPs. Remember, I started with about 1,600, narrowed it down to about 28 or 30, and then was able to identify just those handful that were adaptive and helping the organism survive. The second type, targeted sequence capture, the difference there is that you're isolating a very specific region of the genome or a few regions and then using high throughput sequencing to only sequence those target regions. When we're thinking about the potential benefits of this technique over the other options, reduced representation or whole genome sequencing, which we'll talk about later, one of the benefits is that you get really deep sequencing of that one region you're interested in. And that's in contrast to these other methods that have more shallow sequencing spread out across the genome. Deep sequencing just means you get a lot of sequence reads from that same genomic region. Another advantage for targeted sequence capture is that because you're only sequencing one particular region, it's easier to analyze the data bioinformatically. So it's not as intense to figure out how to analyze it like whole genome sequencing is. And then what makes sense, because you're only sequencing one portion of that genome, targeted sequence capture is cheaper than whole genome sequencing, for instance. Okay, and then the third type, whole genome sequencing, is where you're sequencing everything, results in the DNA sequence of the entire genome. And they've actually done this. They've sequenced the whole genome of the giant panda for conservation purposes, and they found some really interesting data. Like, for instance, the panda has mutations in one of its genes that they think means it can't taste meat. And so they think that's why pandas eat bamboo, even though they're bears, instead of meat, because they actually have a mutation and they can't taste meat. So that's super interesting. That's one example of an organism that has had its whole genome sequenced. For some research questions, it's necessary to sequence the whole genome. So this is an image of a collared flycatcher, and in this particular study, they were interested in, across these different generations, they wanted to know every single mutation that occurred across these different generations so they could calculate an overall mutation rate for this species. In that instance, it was necessary to sequence the entire genome to be able to get that type of information. And as we've talked about before, whole genome sequencing gives you tons and tons of data, millions and millions of SNPs, as was done in the Atlantic silverside, which is a type of fish that's shown here. They identified 2.5 million SNPs. But it is important to keep in mind that whole genome sequencing is really expensive because you're sequencing the whole genome, and it gives you lots of data, which means it requires bioinformatics expertise, both in terms of the person who's doing the bioinformatics and in terms of the computer that you're using to do that analysis. It requires a lot of bioinformatics expertise. So to close out this module here, I want to revisit that example that we started this module with at the very beginning, looking at my little intertidal snail, Cephalnebralis. And we showed at the very beginning of this module that there were conflicting results about whether or not individuals from different populations were genetically similar or genetically different based on these two different studies. And I want to point out a couple things here. First of all, this study was conducted using single gene sequencing. And they specifically used CO1, which is a mitochondrial DNA marker. In my study, what I did is I used SNPs that were identified through RADseq, which is a type of high-throughput sequencing. And another thing I want to point out is the dates that these papers were published. So the first study was published in 2010. My study was published in 2016. So a couple points to make here. One, the technology has really advanced in the past couple years to make high-throughput sequencing much more feasible for these types of studies, which wasn't really the case back in 2010. Another point I want to make is that in general, if you have the money to do a high-throughput sequencing study, it's generally preferable. And the reason for that is that high-throughput sequencing gives you lots more data, which means you're more likely to get an accurate representation of what's actually happening in those individuals or in those populations. So in this example here, when you only looked at single gene sequencing from a mitochondrial DNA marker, the results told you that there were no genetic differences between individuals along the coast. But if you look at that in a different way and instead use thousands of SNPs and narrow that down to adaptive SNPs, you can actually see that there are genetic differences between individuals in Northern and Southern California. And so really that's why I say high throughput sequencing is my favorite method, is that because of the amount of data it gives you, it can give you a really accurate whole picture of what's happening in this organism, and then you can relate that molecular information to what's happening at the ecological level to give you this really, really cool, complete molecular ecology study.